I did my capstone research on pediatric pharmacokinetics and drug dose development. So my thesis looked at the maturational changes that children go through throughout growth and development and how this influences how their bodies process medications. So currently, the most used method to alter an adult dose into a child's dose is to alter it based on their weight. However, ever since basically the early 2000s, it has been noted that this isn't really a very effective way to alter the dose. So to begin, I think it's good to start with looking at the current drug dose development process. So this is it on the screen here at a very surface level. To start developing a new drug, you start in the preclinical phase, which usually involves testing in a lab and on animals. And once you've accumulated enough data to sort of ensure that your drug is safe and effective, you file it through the FDA, and if they approve it, then you can enter the clinical phase, which involves testing on people. So phase 1 involves looking more into safety and dosage. Phase 2 looks at efficacy and side effects. And then phase 3 looks at more long-term side effects. And then once you're done with phase 3, you sort of accumulate all the data you've collected from the beginning, and you file for what's called an NDA, or a new drug application. So then the FDA looks at this, and if they think that your drug is safe and effective, then it can be approved to be sold on the market. I think it's really important to note that the FDA does a really good job of regulating manufacture, promotion, and sales of drugs once they hit the market. However, they play no role in regulating how prescribers use these medications once they're available. And I also think it's important to note that this process is sort of the root of the issue that I'm highlighting today, because it would be extremely unethical to test new drugs on children, and because of that, we lack data on how drugs function in pediatric populations. Okay, so off-label drug use. Because of the fact that there aren't a lot of pediatric formulations, prescribers need to just sort of change the adult dose to make it work for a kid. And this would be considered off-label drug use. So it's using any medication for something or on a group of people that was not specified on the drug label or the packet insert. To get put on the packet insert, it must have enough data must have been given to the FDA for them to include it on there. And because you're using it on a population or an age group that wasn't included, this increased the risk of adverse side effects, toxicity, and a lack of therapeutic effect based on an incorrect dose. There have been a lot of studies, both in America and European countries, that showed that 1/3 of all inpatient children in hospitals receive at least one off-label medication in their stay. So all of these factors combined really raises a red flag for the fact that we need to improve research on forming pediatric formulations. Luckily, the FDA has been doing things to sort of combat this issue. In 2002 and so on, most of these initiatives were enacted, and they all have an overarching goal to increase the amount of research being done on pediatric populations to improve the labels of drugs that are already out on the market. This all sort of started with acetaminophen, which is Tylenol, a very common over-the-counter analgesic. A lot of children and adults were facing severe and acute liver failure because of toxicity by taking too high of doses. Acetaminophen is something that not only is it in Tylenol on its own, but it's also incorporated into a lot of other medications. So parents that were trying to combat their kid's cold would give their kid an array of medicines, and they all included acetaminophen, but they didn't really realize it, and then their kid ended up with some toxic side effects. So they started these acts in order to improve the labeling on pediatric meds. And while these are all really great, there's still a lot of work to be done. So these are just slow steps sort of in the right direction. So now we can get into the fun stuff. So pharmacokinetics is a branch of pharmacology, or the study of drug action. And it follows the movement of a drug from the moment that you take it, in whatever form that may be, to the moment that it is excreted and eliminated from the body. So first is absorption. Absorption basically follows the drug from the second it enters your body, from whatever mode of administration, until the moment it reaches your bloodstream. So there's a lot of different types of modes of administration. There's orally, rectally, subcutaneously, and so on, and they all have various advantages, disadvantages, and purposes. So the one that I outlined on the board is orally, because Tylenol is typically taken orally. So first, the drug is swallowed, and it travels through the throat and the digestive tract until it reaches the small intestine, which is where absorption occurs primarily. From there, it's let into the intestine through these basically like specialized proteins in the intestinal wall called drug transport proteins, and it first moves to the liver before then traveling through to the bloodstream and circulating before it reaches the target site. So there are a lot of maturational changes that go in hand with absorption when comparing children and adults, and most of them are determined by the state of the GI system, so intestinal composition, motility, gastric emptying rate, gastric pH, and volume. To hone in on a specific example, researchers found that gastric pH is really critical in determining oral drug solubility, or the amount of time that it takes from the drug you swallow to go from the pill or liquid form into the active molecule that then continues to travel through the digestive tract. So infants or neonates, right after birth, you're born with a very high pH. It's very basic, and it doesn't reach adult levels until like 2 years of age. And for medications that are weak acids, like acetaminophen, this high pH acts as like a protective barrier, and it slows the amount of time that it takes to break it down into the active molecule that can then be absorbed. Another thing is the intestinal composition. So right here is like a histology image of the innermost layer of the small intestine that faces the hole where everything travels through. And it's full of all these villus folds and crypts, and these are made to maximize surface area for absorption. And these folds don't develop until, they don't even start developing until like 8 weeks after birth, and then it takes a long time before they start to look like this, and that greatly decreases a child's absorptive capabilities, so a dose would need to be altered accordingly. Okay, next is distribution. So absorption followed the drug until it got into the bloodstream. Now distribution looks at the drug moving from the bloodstream into a given tissue. And this occurs at varying speeds and rates depending on the drug solubility, if it's water soluble or fat soluble, as well as whether or not the tissue has transport proteins ready and available to allow the drug into the system. Again, this has a lot of maturational variations that would need to be taken into account to alter the dose, one of which being plasma protein concentrations. So drugs have affinity for these proteins. They bind onto the plasma protein, and then they're not free in the bloodstream anymore. So this would be like a free drug that's ready and able to move into the tissue, but these are supposed to be plasma proteins, and the drug, if it gets bound to that, it stays there, and then it's not available to enter into the tissue and have the therapeutic effect anymore. This is really critical because neonates and infants have like a drastically lower concentration of plasma proteins than adults do. So even if you were to vary the dose based by weight, an infant would have much more of that dose getting into their tissues than an adult would because they don't have as many plasma proteins stopping it from leaving the blood. Next is metabolism. So metabolism is the most complex aspect of pharmacokinetics when comparing children to adults. And this is because of a lot of different things, but mainly enzyme expression and major organ development. So your organs aren't fully developed until you're like 15 or something. So until that point, the liver, which is where primary metabolism occurs, it's not working at as an efficient of a rate as it is in adults. And also enzyme expression. So drug metabolizing enzymes, mainly cytochrome P450 enzymes, are the main mechanism for altering a drug, and that all occurs in your liver. And there's tons of these enzymes, and a lot of them don't even start development until later in life. So researchers found that enzyme CYP3A1 doesn't even start developing in your body until 20 days after birth. And another example would be enzyme 2E1 doesn't become fully developed until a person reaches 10 years of age. So when you're thinking about something like acetaminophen, enzyme 2E1 is used to basically turn off the active molecule. So it takes the active molecule that's entering the tissue and having its therapeutic effect, it turns that into something that's inactive, can be further altered and excreted. So if you're a kid and this enzyme is not present at all, or if it's less present, the drug will stay active in your body a lot longer than it would in an adult, and the dose would need to be altered accordingly. So next is elimination. So elimination is the process of clearing or excreting a drug from your body. Again, this has a lot of different maturational factors. Both revolve around renal function and hepatic function. They did this research study in South Africa where they looked at liver to body size ratios, because even though children don't have as fully of a developed liver as we do, it starts growing to be normal size, to be whatever size it would be in an adult person. So when you're 10 years old, that liver is a lot bigger in comparison to the rest of your body than it is in an adult. And because of this, kids end up clearing and metabolizing drugs a lot faster than adults do. So that needs to be taken into consideration. Another thing is back to these drug transport proteins. They're expressed at varying levels throughout life, and these transport proteins are what allow the drug to get into the organ to be metabolized and altered, and also what allows it to leave and make its way to the kidneys to be excreted throughout the urine. This figure right here is from a research study that looked at hepatic uptake transport protein expression in human livers at various ages. So it did neonates, infants, children, adolescents, and adults. And you can see that there's really varying levels of expression throughout age, and this greatly alters the rate at which your body processes medications. The most pronounced one was OCT1, so it's at a very, very low expression level here, and it doesn't get until its max until you are exactly in adulthood. So that must be taken into consideration as well. Okay, so part of our capstone thesis was to propose an area of further research. So I've been talking a lot about how there's a lack in pediatric data, or in data on pediatric populations, and unfortunately there is no one-step solution to this problem. The best we can hope for is really to start to narrow the knowledge gap and eventually close it between these age-based factors that alter pharmacokinetics. So I chose to look more into these drug transport proteins that I was talking about on the last slide. I specifically picked to focus on transport protein OCT-N1. There's currently no relevant data on how this alters how children process medications. So OCT-N1, it's an organic cation and ergothioneine transporter, and it also plays a role in L-carnitine tissue distribution. So this has some pretty serious clinical relevance because carnitine is necessary for the beta oxidation of fatty acids. And if you were to do something that would alter the beta oxidation of fatty acids, that would slow down the process of getting the molecules into the TCA cycle, the electron transport chain, and slow ATP production. So if another drug were also a substrate for this transport protein with the carnitine, and it would block it, and it would be sort of competing to get through, and the carnitine wouldn't be able to get through as often, this would have a lot of negative metabolic outcomes. So if I were to do this research proposal, I would determine gene expression of the OCT-N1 in various ages, and then do some sort of statistical analysis to see if I could apply this data to like a current model of the human physiology in order to predict how it would influence drug disposition. So I would take tissue samples, probably of the intestine. OCT-N1 is expressed in both the kidney and the intestine, but because of the oral mode of administration of acetaminophen, it would probably have more influence in drug absorption in the intestine. So I would take tissue samples from mice at various ages all the way up until adulthood, and adulthood for a mouse is considered to be about 8 weeks. I would use some sort of kit to extract the total membrane proteins from these tissues, and then I would run it through a liquid chromatography system that was tandem to a mass spectrometer. So the first portion of it, liquid chromatography, would basically run these samples through a teeny little column and separate the proteins in the sample based on size. And then a mass spectrometer basically blasts the samples with electrons to break them down into smaller ions. And then this data is spit out, here, it would look something like this. So the data is spit out and it breaks it up based on like mass to charge ratio. So each of these peaks would show you a different protein. And then the size of the peak, or the peak's intensity, would tell you how much of that protein is in the specific sample. So you would look at how much of OCT-N1 was expressed in each age sample and do some sort of statistical analysis to determine if there was any age-based expression. And if there was, the hope would be to create some sort of formula that could be able to alter adult clinical data to include how it would change if it were functioning in a child. And then this would be included in some sort of pharmacokinetic model to predict outcomes. So that way you could use adult data to turn it into something that would be